This is Nanoscape. I'm Seth Zimmerman.
And I'm Erin Spain.
On this show, we venture into the frontier of nanotechnology
with experts in the field who are turning the science of small into the next industrial revolution.
This podcast is brought to you by the International Institute for Nanotechnology at Northwestern University. Today's guest is Nathan Gianneschi, who has been called one of the most creative and energetic researchers in the application of nanoscience to medicine. As a Jacob and Rosaline Cohen Professor of Chemistry at Northwestern University, with additional appointments in the departments of Material Science and Engineering, Biomedical Engineering, and Pharmacology, Nathan embodies an interdisciplinary approach to nanomaterials research that has the potential to significantly impact human health.
We welcome Nathan to the show today to discuss his lab's work in biomedicine, nanoscopy, soft matter, and biomaterials. We'll also get his insight into some of the hottest topics in the field of nanotechnology today. Welcome to the show, Nathan.
Thank you very much, Erin. Thanks for having me.
Our listeners may detect your New Jersey accent today. So tell us about your background a bit and how your career and interest in nanotechnology brought you from Australia to the States and here to Northwestern.
Yeah, no, I've been to Newark Airport. No, I grew up in Australia. I was born in Canberra, which is the capital city, as some people may know, and then lived for a time in Melbourne. We lived overseas for a while, traveled a bit as kids, and ended up back in Adelaide, which is in South Australia, where I went to the University of Adelaide, where I majored in chemistry and biochemistry and did my undergraduate research in chemistry, in particular in inorganic chemistry. It was at that time when I was thinking about going to med school, like I think my parents might have wanted me to, and I certainly had said I wanted to. And then as I got into laboratory research as an undergraduate, both prior to my honors degree, you do a 3-year degree in Australia, plus almost like a mini master's degree prior to a PhD. I was doing that with Professor Lou Rendina, who was actually starting his career at the time when I was in his group. He was very interested in nanoscience, the possibilities. And this is 1997, 98 when I did my honors degree. He's handing me papers and manuscripts, as your professors often do, and saying, you could do all these things. You could change the world. And so for some reason, I believed it. And I think maybe I still do. But, I got to get in the lab. And without knowing much chemistry, just sort of undergraduate degree level type work, we started making new molecules and started seeing new phenomena. And that got me hooked, literally. So I decided, reading all these manuscripts and thinking, I want to do research for my career, for my life, I should come to the United States. And that was partially prompted by Lou. And this is against the dogma, right? You're meant to keep your undergraduates in your lab in Australia and not send them overseas. And I decided to do that. I ended up moving to the United States, to Northwestern, to do my PhD.
Was this a difficult choice for you to come to Northwestern, or was this the place you wanted to be?
It was my first choice, and I applied to several other schools, some of which I got into, some of which I didn't, which is the way it goes, of course. I was working in the lab actually late at night. I think I was drying hexane in a still. Everybody else I knew was at the pub. And I got a letter from Northwestern saying that I needed to take the English as a second language exam because I'm a foreigner. And so I'd written a note to the graduate assistant at the time in the department and said, I'm more than happy to do it. Really sorry I missed this. Are you 100% sure that I need to do it? Because I'm Australian. And he'd either called me or emailed immediately. And he said, no, we got that wrong. You're in. I mean, that was a pretty happy moment. I immediately, of course, turned the still off and went to the pub. But no, I was very excited about it. And ultimately, that was really driven by the people. And so I had applied here because I wanted to work for Chad Merkin. I'd been reading papers where they'd been looking at a range of things. And that period of time, they were doing the first sort of work on modifying gold nanoparticles with nucleic acids. They've done a lot of that work, and others have followed up on it. So now it seems like a normal thing to do. But to me anyway, it was quite a weird thing to do. Here's a biological molecule, and here's an inorganic metal nanoparticle. They've got nothing to do with each other. They're from different planets. People who study one don't study the other. And that was turned on its head. And as I said, other people are interested in that interface. So I was interested in bio-inorganic chemistry, but more from the perspective of studying metalloenzymes, not really in the nano domain. And that really changed my mind. And I ended up working more specifically on organometallic supramolecular chemistry with Chad.
And so you were here at Northwestern, you finished your PhD, you went on to some other roles, but then you came back to Northwestern. Tell me about that decision to bring your group back here and what you all are doing now. What are you studying to use nanotechnology to improve human health?
So I went to Scripps in La Jolla, which was a fantastic place, and did a lot of interesting creative work with Professor Reza Ghadiri as a postdoc. Started my lab at the University of California, San Diego. We really went in a new direction from what I'd done as a grad student, as a postdoc, and got interested in polymer science, soft matter, and using soft materials approaches to develop nanomaterials. And so that flavor of project is ultimately what we brought back to Northwestern. San Diego is a difficult place to have lived in and left. I was saying the other day, no matter where you are in the world, the weather is better in San Diego. We'd had the discussion ongoing about a possibility of being part of the International Institute for Nanotechnology, being a member of multiple departments. Obviously, I'd stayed in touch with people here over the years, had visited several times, and then visited specifically under the guise of an opportunity to come. And so, yes, it was difficult to leave, but it wasn't difficult to be here at Northwestern, especially because then I had appointments in multiple departments, all with interest in nanoscience and material science, biomedical application of material science, which is what we do, and then really close connections and opportunities with the med school as well. One of my graduate students to move with me, who first graduated from here, Molly Tuvey, she was an electron microscopist. We'd always struggled to get access to instrumentation at UCSD just because their focus is different. Northwestern has some incredible facilities, some of the best in the world, for doing nanoscale characterization in particular, which is a big part of what we do. I said, Molly, how was it? She was from California, right? She'd grown up in California. She was at UC San Diego, and I dragged her to Chicago. And she said, well, I wish you could have moved Northwestern to San Diego. So I often tell people that because, of course, it's obviously impossible, but it tells you a lot about how even our Southern California kids came to appreciate the kinds of experiments we could do here. And I sort of use that to go back to what you asked about. It's the resources and being able to do almost any experiment you want. Having your own ideas be the limitation is a massive, massive thing. And it's not just Northwestern, it's the region, Argonne, et cetera. And we can ask scientific questions and through collaborations and also through direct training of our students that would have been very difficult and were very difficult for us to do before, but have also opened up new directions, new questions, new problems, and things like that.
Some of the work that I've read about that's coming from your lab includes synthetic melanin engineered nanoparticles, which are designed to heal the skin from a variety of injuries, and proteomimetic polymer technology, which is a promising therapeutic strategy for a range of diseases, including neurodegenerative conditions such as Alzheimer's, Parkinson's, and ALS. Can you tell us a little bit more about these projects and what makes these conditions such a challenge to treat?
We became interested in melanin, which is a skin pigment in all of our skin and hair, and depending on our skin color, we have different amounts of it. Primarily provides protection to our skin from damaging UV light, but also damage from environmental toxins. People have become more familiar with it in recent times. We got interested in it from a basic science perspective in about 2014, so we've been working on it for about 10 years now. I like to say it's ubiquitous and enigmatic, so I just mentioned it protecting our skin from UV light. It also protects other organisms from the same kinds of damage, and yet it's found in deep-sea fish that are way beyond actinic radiation. So in other words, they're not at all influenced in the natural environment by UV light. In fact, they don't have any impinging sunlight at all. So it's used in that case as a thermal regulator. We figured out together with a large team of scientists, an Air Force-funded program, a basic science program, what is melanin from nature, what is its biology, what's its chemistry, and then really excitingly, what's its nanoscale properties, and how does its properties at the nanoscale give rise to other features. So for example, thermal insulation, but also structural color. So peacocks are completely melanized. Their feathers are iridescent color that comes from a black pigment but gives those colors. Many hummingbirds are the same way. And this has to do with particle shape and the way that the particles are positioned with respect to each other, sort of the chemistry through to the nanoscale, through to the microscale organization of matter. And so that's a very interesting topic way beyond melanin as to how we create structural colored films or architectures that could influence, for example, the way you might paint surfaces. So they don't get bleached, let's say, or damaged by a photo. They are more like physical structures. So there's a lot of interest in that space, way beyond melanin. through that process, we got to really dive into the basic chemistry of it. How are they made? Can we mimic how they're made? And we figured some of that out. There are other labs in the world who've figured other parts of it out. And this led to a meeting of two worlds again, like many opportunities in nanoscience in particular. It's very interdisciplinary. And you'll probably hear that again and again as you talk to different people. It really is at the intersect of a lot of different fields. In this case, strangely enough, between chemistry, material science, polymer science, nanoscience, and dermatology. And so I was invited to give a talk in the medical school at dermatology. I presented about what melanin is, how we use it. We can put it in fabrics and all this kind of stuff. A couple of dermatologists, Robert Lavka and Kurt Liu, who were running the show that day, came up to me and said, have you thought about this in terms of putting it on the skin to heal the skin post-injury? So if it's useful inside the skin to heal from injury, could we develop something that would be a topical? And I think we all thought, let's try it, which is a great collaborative attitude, but it probably won't work. So we got preliminary data, we got research funding from the NIH, and we built a program together with a whole bunch of other researchers with their own nanoscale particle systems for skin healing, some injectable, ours topical. And Kurt Liu directs that program. It's a U54 grant at Northwestern that includes material science and Feinberg. It's almost like exactly what you would want and exactly the kind of thing that's possible at Northwestern by really allowing those different departments to work together. And so once you get money, you get grad students and you can start doing stuff. And it turned out to work. We've been able to apply it to skin injury after UV burns, after chemical burns. We're working on radiation injury. So these are clinical radiation burns that are caused by gamma treatment, for example, for tumors. Most patients acquire radiation dermatitis from those procedures. Their skin becomes inflamed, etc. And there's no treatment for them. So we're particularly excited about the potential to use really a precision nanoscale chemistry and synthesis, control over materials at that length scale, leveraging all the characterization tools, and obviously 10 years worth of work in doing that, but then working with immunologists and dermatologists like Kurt to implement it and ask the really robust biological questions.
So that's been a really exciting example of moving work from basic science through translation because of these Northwestern connections. Can you tell us a little bit more about your work with protein materials?
So our group, the connective tissue here is that we've been interested, as I said, in how polymer science can be leveraged to make functional polymeric nanoscale materials, but also other kinds of materials. We had this idea actually probably even as an undergraduate, and I think many chemists have this thought of mimicking natural systems. In particular, I was interested in whether we could eventually mimic proteins or enzyme active sites. And there's an entire field and a whole lot of literature in that space. So that was sort of in the back of my mind. And we moved into these projects using polymers labeled with peptides. So we can display peptides on a synthetic polymer scaffold. And we were working actually on a different kind of project. Serendipitously, we found that that particular form of matter has all kinds of interesting features. It gets in cells. They can bind proteins that other molecules can't bind. So they behave in ways that are totally different to antibodies, totally different than small molecules. Because of their size and shape, their properties, they circulate for long periods of time in vivo and have allowed us to go after lots of different targets, which you asked about. Neurodegenerative disease, retinal disease, which we work on together with Jeremy Levine in particular from the medical school and ophthalmology. Those efforts also led to a startup company with Portal Innovations. So we have laboratory space in the West Loop in Chicago. So it's a Northwestern startup with a whole lot of obviously licensed IP, and they're out doing Series A right now to take those materials forward into the clinic, particularly focused on cancer. Our research program is on the basic research side, where we continue to ask questions about mechanism and can you continue to use polymer science and nanoscience to mimic proteins, maybe other biomolecules, collections of biomolecules, and then how far can you push this in terms of mimicking cellular components? And so there are bigger questions there and implications for how we might think about mimicking cellular structures or drugging specific targets using kind of a knowledge of how to precisely position, or as precisely as possible position atoms within nanoscale structures.
You're really part of what's being called this nanotechnology healthcare revolution. Can you just speak a little bit about this broadly? What's happening right now in this field when it comes to nanotechnology and healthcare, and what energizes you and excites you about this?
I think it's like other times in history where you've got this wave that's kind of starting to sort of break. You've got some momentum behind this in terms of a lot of different background technologies coming together to enable what the outside world will see as technological outcomes, so real innovation in the marketplace. Nanotechnology enabling specific kinds of television sets to be made. But that's leveraging decades of work on the basic science side. So you're starting to see the results of a big investment, private, public, multiple governments around the world who really maybe took a risk in the late 80s and early 90s and until now behind what was not a field. It was a collection of people saying that there's going to be something important at these length scales, and that's now coming to fruition. I think in terms of the concepts behind nanoscale materials being useful in medicine, where a lot of our work has been, you see this in great examples. The COVID vaccine, in particular, the Moderna vaccine and the Pfizer vaccine were both developed using specific types of nanoscale materials that are lipid composites with nucleic acids. I think that's opened that there are limitations of that in other diseases, so systemic delivery and things like that. But as a field in what's called nanomedicine, people are starting to come into grips with the limitations. I always tell my organic materials class to graduate students at Northwestern that if you understand the limitations, you may have a chance at understanding the opportunities. And I think the field is starting to see part of that discovery curve. Where will these things really be good, as opposed to where small molecules are good and where antibodies can be great? So where do we have opportunities? Where is there white space? In particular, in our case, an interest in human health, but I think you're seeing this across the board.
I'd like to touch on something that you kind of mentioned, how nanotechnology impacts other sectors beyond medicine, such as electronics and environmental science, which I'd love to hear a bit more about, because that's fascinating to me. Where else you can see this beyond the medical world?
I think you see it in anything in chemistry, nanomaterials, nanoscale features. So not just, say, an object that might be a sphere 100 nanometers across, but also a feature, a top-down approach to making patent materials, a lot of where we're trying to go with modern electronics. The other way around is mostly what we've been focused on, which is bottom-up. I mean, as chemists, we make molecules, we try to control them, then we make polymers, which are a little bit less controlled, and then they assemble into larger structures. Even the lipid nanoparticle that I mentioned is the result of an assembly process. So I think the challenge in there has been just exactly that. How do we predictably make objects that have a specific function in a reproducible way when you're not using chemical bonds to make them? And so this is sort of the chemistry beyond supramolecular chemistry that then leads to these larger length scale interactions. You mentioned environmental chemistry. There's been a lot of work in the influence of aerosol particles on cloud formation. These are fields that I'm peripherally aware of. We've worked with and collaborated with some people in those spaces. But there's a lot of effort, right? And our awareness that those are important properties of matter trails our ability to characterize them. And so one of the things that I've always been most excited about is our ability and the inventions over the last 100 years in first molecular characterization, but then ultimately nanoscale characterization. If I can measure something, if I can see something, a nanoscale defect in a material, if I can detect a nanoparticle and describe it fully using electron microscopy, then I can study it. I could modify it as a chemist and I can go from there. So those real enabling inventions that we are now reaping the benefits of, and in our own work, optimizing, developing new modalities, but basically allowing people to characterize materials in a better way. I think that's where some of the exciting advances are getting their legs from.
Now, you mentioned a lot of this is just years and years of development that have now led to what feels like at sometimes nanotechnology moving at warp speed, especially in nanomedicine. There's so many papers now being published every day in this area. But along with this comes some legal and ethical considerations related to research, development, and use of nanomedical technology. How do you and your colleagues approach these issues related to legal and ethical concerns?
We view a lot of, I think, what you're asking about through a lens of, let's say, an FDA approval path. And so there's obviously a lot of questions in it. It depends on the exact particle. It depends on the way it's being used. But these are questions that have asked of new small molecule drugs as well. Biologics that should otherwise be fine, right? If they're a new chemical entity, if they're a new material, they've never been applied in a human, you begin in your lab with testing toxicity in cells and you go from there. And that whole line of thinking, if I'm going to do something novel to perturb a cellular structure or a process, I better make sure I don't perturb other things. Do I have on-target toxicity? So is the toxicity I'm observing because I'm overstimulating some pathway, but I'm on pathway, and then it's a question of dose. And so the answer is that we take a very detailed look at this at every level from the moment we make the new material. Can you aerosolize it? Could I breathe it in? How do you handle it in the lab? Our labs are in controlled environments. We have fume hoods, and chemistry itself is very self-regulated too. Our students are trained from day one to wear protective equipment. And so you are trained to treat almost everything as if it's dangerous, because sometimes you don't know. And so if I don't know what's dangerous, then I treat everything as if it is. I mean, that shouldn't prevent you from working on it within reason. As you said, there's a huge body of literature. And so you can have a bit of a guess as to which compounds are toxic. And I can tell you in the translational space, you start talking to certainly NIH, but way beyond that, investors, inexperienced and experienced investors. They'll ask that question. Is this safe? How do you know it's safe? Where's the risk? Immune risk, so immune tolerance, either to destroy the drug's activity or to create a negative reaction from a person. Those are things that are constantly being tested for all the way up the chain. You can see this even in the initial safety trials at phase 1 clinical trials, for example. Probably where it's less looked at is in other kinds of exposure from particles in the context of, say, if I'm recycling an electronic device that contains nanoparticles, do they get into the groundwater and other kinds of environmental effects? There are labs who look at that, right? How do batteries end up contaminating the ocean? There are people who study those types of things. These days, there's a lot of interest in not poisoning our environment. Maybe it was ignored at least the first part of the last century. But on the side of the world that we're in, it's like day one. And I'll tell you, some investors will even say, well, I don't know if it'll ever be safe, so don't work on these kinds of nanoparticles or those kinds. Particles should be this size and not that size, because there is some literature, right? Some of those are assumptions. All of it needs to be tested.
It kind of made me think also about like when the COVID vaccines were coming out, people thought, oh, this clearly got rushed through the process. And what is this? We don't trust it. It must be brand new. And it wasn't brand new. People have been working on this for years and just happened to be like, okay, well, we have a chance now to roll this out.
I really like this example. Again, I teach it to my class. And so if you look at the history of those types of particles, they were being published, being used on Zika. They'd been tested in all kinds of model systems. Those companies didn't wake up in February of 2020 and just make that up out of the blue. But they also went through all those kinds of safety testing. It's certainly expedited, as we all know, a remarkable example of how fast things can get done if you got enough money and enough motivation. And so I think it rightfully brought up some questions about, hey, how fast could we really make this stuff go? Some things just take time, right? There are certain types of models in neurodegenerative disease that can take months and months and months. There's just no way to speed it up, and some models are faster. But all of it has to go through that process. As an inventor, you can imagine what you set out to do is to help people. The last thing you want to do is do the opposite, right? Could we make mistakes? Yes. And as scientists, of course, you try to get an idea of the percentages, right? And where the risk is, and as early in the process as you can. Sometimes it isn't toxicity. It's that it doesn't dissolve. It's not soluble. You can't safely inject it. There can be physical characteristics, not just of nanomaterials, but of any chemical that prevents it being useful. Those can be sad moments for researchers, and I'm sure that's true for companies as well. But all of that's in the testing. Can I dissolve it in human blood? Can I dissolve it in saline? Can it be put in a bag? Can it be stored for 3 years? Does it aggregate over time? And can a nurse easily redissolve it at point of care? Can it survive 40 degrees centigrade if it's sitting on a tarmac in Melbourne, Australia, which we've had happen to samples? There's a lot of that hard work that goes way beyond the initial few fancy experiments we do in our labs. And I think that's where there's an increasing interest in doing that, at least from us, especially for some of these technologies that we think have got to the point where the basic sciences, we've done enough to know that the next steps need to be done more in the private investment type world and more in the translational space, especially to go to that quality, right? You're not at prototype stage. You're building Ford Model Ts now.
Clinical trials.
Yeah.
You're on to the next step.
So you need a real process. And it's been an education for me. And I'm happy that I have my lab because we can still go and invent some new weird thing. But I think always with a mind to potential utility, safety, and those things.
Something you touched upon is you were encouraged to come to the United States from Australia. Do you see that as a common occurrence? We're the International Institute of Nanotechnology. We're an international collaboration. So we have people from across the world. Is that kind of the case for everyone that you've met so far in your career that like, okay, you reach a certain point this is the place you want to be. This is the top of the nano food chain in a sense that you want to come over here? Or is there other parts of the world that are doing some amazing research that people should be considering?
Certainly, I think Northwestern has done well in attracting talent, and you can see that in the numbers of people and the kinds of people who've moved here over the last few decades even. There's a lot of reasons for that. I mentioned equipment earlier, but the piece of equipment is just a paperweight unless there are collaborators and colleagues and graduate students who can ask the right questions and utilize them. But I think one of the great strengths in these institutes in the US in particular has been that they've been able to attract so many people from different parts of the world and different parts of the country. There's this kind of crazy culture in America in academia of moving. I finish my undergrad, I move. I do my PhD somewhere else. I finish my PhD, I move, and I go do my postdoctoral fellowship somewhere else. If you stay in academia, obviously if you go work for a company, you are leaving, right? But there's this fearlessness about moving around, and we get this percolation of ways of doing science, of beliefs perhaps, certainly scientific training. And so a lot of us in these departments, these kinds of programs, greatly benefit from that idea that there are students in Korea, there are incredibly well-trained students in Europe, all over the world, in China, who are willing or able or excited to come to different places. And colleagues too, right? We'll recruit senior colleagues from other parts of the country and other parts of the world. I think we benefit from the fact that other countries, other communities, and other highly competitive universities all throughout the United States are also training their students in these fields. If we were just an island, then you wouldn't have this. Science operates much more as an international and national community than it does as an internal looking one. We look outward to the rest of the world. What are other people doing? Where are the big problems? Where are the unmet needs in the clinic, not just in the United States, but all over the world? And how can we have an impact on that?
Well, as we wrap up today, I want you to look into your crystal ball and just tell us, what can we expect in the next decade when it comes to nanotechnology and the role that it's going to play in improving human health?
Yeah, that's a really big question. I sort of touched on this. I think our biggest challenge in this space, maybe thinking specifically about bottom up, nature shows us the example that you can have exquisite control over incredibly complex systems by controlling molecular, supramolecular, and ultimately nano and microscale assemblies of molecules. We often turn to nature, especially as chemists, people interested in this space, because we know it's possible. We know that you can have replicating molecules like DNA. We know that you can have what appear to be completely disordered proteins, and yet they control all kinds of complex cellular processes. We're fascinated as a larger community of scientists by that fact and can immediately see the gap. We humans are sort of bound by the fact that we can study these things. We know the gap between how it can be done and how we do it. And we're just getting to tinker at the edges of it. And there are so many people interested in it, right? That's one of the exciting things, from structural biologists and, as I said, cancer biologists who, they're not literally directly interested in nanomaterials per se, but they are actually studying nanoscale phenomena. And the more we learn about it, as I said, the more we know about the gap between what we can do synthetically through, say, human-directed control over nanomaterials and where it goes. So I think the crystal ball thing would be that we continue to be inspired by that and say, if I can understand these networks, which are collections of molecules, collections of nanoscale materials, and how they define or drive human disease, if I can understand that and mimic it, then I can develop drugs and potentially therapeutics. I think we'll get there. I think we're decades away from being able to do that in the really exquisite way that nature does it. It won't happen one day, right? There'll be incremental advances that will result in big jumps when we get to transformational inventions, which we see all the time, and as we've talked about. And you've got to keep an eye out for it. I mean, it's sort of one of those things, I guess you might say at the end of a podcast, like, watch this space. But I think certainly we do, right? And we try very hard to offer new directions and pay attention to those being made by our colleagues and collaborators.
I think that's a great note to leave on today. Thank you so much for being on the show today.
Thank you very much for having me, Erin. And it's a real pleasure. Thank you, Seth.
Thanks so much for listening. This podcast is brought to you by the International Institute for Nanotechnology at Northwestern University. Be sure to follow this show on Apple Podcasts or wherever you listen to podcasts. And be sure to give us a 5-star review.
For more information about IIN, go to our website, iinano.org/podcast.